Hey there, you're about to watch a recording from one of our online events, and in there I discuss with Sriram about some misconceptions about starting your journey in sustainability. We share our personal stories and experiences to help you get started and overcome all of these challenges. We edited it a little bit to make it more engaging, but now without further ado, we hope you enjoy. At first, welcome you all to sustainable practices every scientist can take with limited time and resources. And to basically start all of this out, I want to share with you a few assumptions that we do or have. And the first one is there something like an absolute sustainability? Can we be absolutely sustainable? Because I think for many people want to start, this is always a consideration. And we don't think so. We can be more sustainable in the lab, but it is hard, if not impossible, to be absolutely sustainable just because our research will always have a footprint. We need chemicals, we need energy, we need single-use plastics in sterile environment. So there is no way to be absolutely sustainable. However, of course, we want to become more and more sustainable. And I think this is why we should start here. It is our goal basically to address the thinking of ours, because we feel that we could give you a whole list basically of actions, but what you also should really consider is that you will face many challenges. It definitely won't be easy, but certainly it is possible to become more sustainable. And thus, today we want to unravel the one or other misconception about the field and then basically provide you all the insights into the doing in the other sessions we have, but also what you find on our website, what you will see in our conference, what you will see in our educational lessons that you can get every week. Basically, we really want to link these two together because we think just one or the other probably won't be enough right there. The first issue I think most of you will face is the all in or nothing. It is like what you see on the very left. Like this is sometimes how we feel in our laboratories in terms of waste and organization and opportunities to change something. And on the right is, of course, what we are dreaming about. But as Sriram and I will discuss today, it is not just about being perfect or, again, absolutely sustainable. It is a journey. And of course, there are simple, easy, and still impactful steps that you can certainly use to start with all of this. We thought it would be interesting to share our stories. And as I told you already, I was, so to say, still a baby in the lab. I started during my bachelor thesis, and I was working with cell culture and protein science. When it really made click for me was when I realized that at home, I did so many things to be sustainable, but right when I entered the lab, it all falls off. I'm basically a different human being who's not thinking anymore about sustainability. And I know from you, Sriram, it's pretty similar, right? It was a pretty trivial thing that basically led you.
Absolutely. Yeah, I was just listening to a YouTube video where someone was saying how by just simply shaking your hand 10 times right after you wash, just a small piece of paper towel was enough. And by covering all the surfaces, you could just entirely dry your hand with just one piece of paper towel. It was amazing that, my thought process, of course, led me to the lab where we're used to always pulling multiple Kim wipes or multiple paper towels for a small spill. And then I said, well, why not I just try using one Kim wipe? And then it worked. And then one thing led to the other. I mean, now I have like a paper towel that I always have on my bench. And then if it's a spill that's just water, I wipe it and then let it dry and keep reusing. And then I've now extended it to gloves, and whenever I go to bathroom, I now carry a handkerchief, and I just dry my hands with a handkerchief, and then every day I just throw it in the laundry, and then at the end of the week it gets laundried. So one thing leads to the other. It's really like Patrick said, one small step, and then you realize how it builds on it, it grows on you, and then you take inspiration from what you're able to do to build on it.
If you look at the bottom, you will see momentum and feasibility. At first, start with something feasible. Both Sriram and I started with very simple and trivial things. And you might not feel like you will save the world right away, but this is not what it's all about, right? It's about building momentum and to get started to do something that you will actually feel comfortable with. The impact I put there because lots of people will always say, but this doesn't really have an impact. Like if I see how many tons of carbon dioxide are produced every day, if I see how much waste basically in the supermarket is just, basically ending up in the ocean. We might completely agree with you, but just think about, for that matter, if everybody, instead of going to work by car, would take, a bus or the tram or whatever there might be, you might automatically have about, like, if they do it once a week, and a reduction of basically 20% of the carbon dioxide that your cars will release. And so Sriram and I thought, there might be three interesting things to discuss here. And I would like to start right away with throwing caps into the recycling trash. I was working in an immunological lab, and so we would basically take the organs from mice and we would basically prepare them. And we would use for this these 15 ml tubes, and we would right away take the cap off because we would never need it anymore because we had strainers on it. And normally you would throw them into the trash. It would be, in the end, sterilized and then, of course, incinerated. All of this plastic would be burned. It's not a big thing to just throw it into recycling bin.
Creating master mixes is something I always do. I realized I save not only time, but if I'm doing 10 PCR reactions and I'm using a tip for each thing, I'm always wondering, have I contaminated this? And I always change my tips. Rather than that, for same reagents, I create master mixes that saves me both time as well as the consumables I use. Absolutely. That's also a low-hanging fruit. If you've not tried it, I highly suggest you try that.
And I think this is leading us to the next thing, the problem of time, because I think we're all researchers, we're all busy. I think very few of us will ever have had a day where it's like, oh no, I did everything I wanted to and I can completely relax. Normally we are already ahead in the next day because there's something we didn't really finish. And still, for me, the breakthrough came to some extent to see it's not just time, it's also the effort, right? It feels like if you don't have time, you're just worn out, you're just exhausted. And this then also leads you to, I can't be so specific to just think about sustainability. Now I really need to focus down on my research. And you see, it is really just about realizing that you do this on the go. And we will come back to this in just a second, but it is just you moving through. It is you basically changing nothing about your experiment in a big sense. It is really just to say, okay, I don't throw the cap in this trash, but in that trash. I prepare master mix because I'm faster.
I mean, the way I figured out a lot of things I can do is like with this example, I watch people do competent cell preparation. When I was doing that, I noticed everyone was always reaching for those consumables like pre-packaged, commercially sold tubes for centrifuging, for example. So it comes at a premium. I mean, what I was able to do was by planning ahead. So when I'm autoclaving media, for example, I'd say, okay, let me autoclave some glassware that I'd use in the same procedure, so I won't have to reach out for those plastic commercially sold items. So by doing that, I realized that I always had a stock of autoclave stuff, and when people started seeing what I was doing, in fact, sometimes they would come and borrow autoclave stuff from me. So by planning ahead, I was not only able to save on, stuff that was commercially sold plastic, using glassware instead of that, but I was also able to mentally run through the experiment, and I realized how efficient I was in completing the experiment on time as well.
I think this is a big point, because if we go back to this time, effort, specificity, this is what I want to talk about here, implementing through abstraction. It sounds so very technical and somehow weird, but I think just what Sriram also mentioned, you take a little bit of time before, but it is not really that you need to go through every single protocol that you have, through every single step and take minutes to figure out what you can do. It will come with time, right? Abstract what you should do or what you want to do in terms of the 6R: reduce, reuse, recycle, rethink, reject, and repair. Especially the reduce and reuse, I think, are big points here. And then you will automatically see basically translate on the go that, oh, here I can reduce again. Oh, here I can reuse my pipette tip because I'm just pipetting water or a solvent that basically ends up in all of my samples. Let's say you want to scan your sample in microscopy. You would normally put your sample into the microscope and say, okay, let's scan, and then we see what's coming out. Of course, if you do this and you might not choose the optimal setting, you have to do all of it again. That means 3, 4, 5 hours completely wasted. And this is your time, but it's also the time that the microscope is on, that the lasers are on, that the computer is running. And so you can really save a lot of time doing this. This also brings us to the last problem on the way of starting, which is, can I really change this? Can I really change my settings? Can I change the eluents in my HPLC or whatever it might be? And I think here we really run into the question of sustainability versus data, or data quality for that matter. I think many people have the inherent assumption that somehow it's like a balance or a trade-off, like you trade the one for the other. Yes, for me, basically the insight came when I was working again in the cell culture. I'm gonna think about what can I do differently. I would still, when cleaning all the consumables I take into the hood, I would still spray them 4 or 5 times more than maybe necessary. It would really drip. And you can say this is not the most sustainable practice, but I would still say, instead of getting a contamination, probably this measure is still very much feasible, sensible. Nevertheless, when you're then in the hood, it doesn't mean that you can be sustainable. It is not about changing the deeper, so to say, scope or goal of your experiment, but rather really how you go about it. It means, as we've had just a few slides before, how you pipette, because in the hood still you want to be sterile, no doubt about it, but how much pipette tips you need to be sterile is completely up to you. I always wondered whether I can, when I just passage my cells, can take the very same plate again, the very same Petri dish again. And here it was all about, just throwing in some controls. You use the same if you just passage them. You might think, okay, but then I will use 1 or 2 Petri dishes more because I want to test it out at first. But think about the long term, how many Petri dishes you will save. And this goes back to the low-hanging fruits. You might think, oh yeah, I want to have a big impact, but how should I do this with small steps? If you do it from now on, you have the next 5, 10, 20 years of your research, and you will create an impact. I think this is something very powerful and that, again, people often forget. I talked about cell culture, no to you, Sriram, because you will talk about radio labeling, right?
Yeah, exactly. So talking about a game changer, one of the game changer for me was, my PhD thesis project as well as postdoctoral work involved doing a lot of assays. So I would run these kinetics assays, which would be 100 to 200 reactions per day. And for every reaction, I was using about 5 mLs of scintillation cocktail. And it was not just me, there were other people in the lab doing similar assays. And, we were realizing we were going through 20, 40 liters of scintillation cocktail per month. So there was this one time when someone from a company came and said, hey, we have a biodegradable cocktail. I know we are not as big as PerkinElmer, but do you want to give it a try? I'll give you a free sample. Not only is ours less expensive, but we are committed to sustainability. So why don't you try this biodegradable one? So it was just a matter of me running a very simple experiment, comparing the cocktail that was traditionally used, that always worked, versus this new cocktail that is now biodegradable. And I realized the counts I was getting for the C14 labeling was the same in both of them. And then I tried out a kinetics experiment. It was the same. So within no time, I was able to convince everyone in the lab, not just in our lab, but other groups doing radio-labeled assays, that this cocktail, the biodegradable version, is just as good, and you can save a lot of money. And guess what? My advisor was super happy that we were able to save a lot on spending the money on cocktails, which can be pretty expensive. If you have worked with scintillation cocktails, you know what I'm talking about.
And this, I think, is a very important insight, because as the picture should suggest here, which path do you take, right? People say never change a running system. It is true you shouldn't do it mindlessly, but it certainly is possible, especially with such simple steps as those. And so again, if we might be able to rephrase it, sustainability and science are really the core foundation, so to say. And what we will trade off here is the optimization versus the convenience. Because many of the things we do are just convenient. It is more convenient to not think about alternatives. It is more convenient to stick to what you're doing instead of thinking about how to optimize stuff.
Yeah, so one of the things I wanted to touch upon was the collection tubes. I realized Kaizen, I'm sure a lot of us, you go through Kaizen kits, multiple kits, sometimes during a week for DNA purification, for example. I realized, the collection columns that you use, pretty much it's not like you're going to elute any final DNA from those collection tubes. All they're meant for are just collecting the washes and other things. So there's no reason you can't reuse those collection columns. And I realized Kaizen just sells just the binding columns separately. So if you wanted to reuse, they do have just the columns for a much cheaper price that you can buy, and then you can reuse the collection column.
I want to take on the shutting the sash because when it comes to working sterile, I think there are many prejudices. And still, if you just, when you leave the bench, shut the sash, you can save a lot of energy. I think there were a few studies now showing that over the period of a month, you can easily save up to 4-digit numbers in money. That means a few $1,000 just because you shut the sash from time to time. I think there's so many of these tips. And again, everybody, you will get the summary like with all of these tips or practices as well. We are about to publish actually a little review where we try to collect everything that we could find throughout the internet and with our own experience in order to be more sustainable. So definitely stay up to date. These are really encouraging stories, but we want you to take something away from it. And this is why I would ask you now to just take, I don't know, open up Word or take a pen and just put one thing down that you want to do. Because if you write it down, I think you will right away have a much stronger commitment to actually do something. I think that would be really amazing. How do I motivate others? When we were setting up all of this, we were actually thinking about whether we should address this question. And maybe to start this whole thing off, my two biggest tips is, on the one hand, share it in your lab meeting, but just in a way that you don't claim that you have the solution or saying we should, just saying that you might think about doing something. Also mention it to your PI. It can be very powerful to say, hey, I've been doing this and that, not changing anything about the experiments, but doing the first things. Do you think that this is a good idea? Do you think that I might be allowed to bring it into the lab meeting next time? I think this is very powerful. And the other thing is, I think numbers, you're completely right, super helpful, especially for older or for more senior researchers, like your PI potentially, numbers in terms of money, right? In terms of saying, if we order this less, you can save money on that. We can all save time on this. But the other tip I wanted to bring in here is to really share how the people can benefit themselves. We found extremely helpful to talk about safety, for example. Lots of PIs, lots of researchers were super happy to say, oh yeah, that is true. If I can change the solvents I use, if I can change the chemicals, we are more safe, I'm more safe, I can work better practically. This is something very powerful as well.
I just wanted to add that if you're wondering, if you're the only one in the lab, how am I going to make a change or make a difference? More often than not, what I found was that, new people coming in are always looking up to you. So if you keep doing what you're doing, even if you're not advertising, if there are newer people joining, they watch you do something in a more sustainable manner, although it may be different from someone else, it's highly likely that they're going to catch on to what you're doing. So you don't even need to advertise. Keep doing what you're doing without, of course, compromising the results. And there will always be a following. Don't worry about it. I've always had that happen. Like I said, I had my stash of autoclave stuff and people started doing the same thing, and they would in fact come and borrow stuff from me so that they would be more sustainable. So good ideas always find their way to.
All right, and this is it. If you enjoyed it, have a look down into the description. There you can find some more information and where to find us.